Hello everybody, my name is Petya Orozova, an associate professor at the National Diagnostic and Research Veterinary Medical Institute, Sofia, Bulgaria. Today I would like to present our study entitled Antimicrobial Activity of the Herbal Product BACTEROMAT Herb against Paenibacillus larvae and Melissococcus plutonius, causative agents of Fouldbrood diseases in bees. This work was conducted at the Experimental Laboratory for Aquaculture and Diseases of Aquatic Animals and Bees, located within the National Diagnostic and Research Veterinary Medical Institute, part of the Bulgarian Food Safety Agency. On behalf of our research team, I would like to sincerely thank the organizers for the opportunity to present our results and to participate in this scientific forum. This presentation is structured as follows. I will first provide general information on American and European Fouldbrood, then briefly discuss current control methods. After that, I will present our experimental design, followed by the main laboratory and field results, a short discussion, and conclusions. To understand how the product works, we first need to look at the diseases we tested it against. The next slide summarizes general information about American and European Fouldbrood. The clinical symptoms of AFB are associated with the infection of the larvae prior to cell sealing, followed by degradation of larvae tissue with characteristic ropey consistency, as well as severe impacts at the colony level and long-term persistence of the pathogen due to spore formation. The causative agent of EFB affects larvae before cell sealing with secondary microbial infections, increasing virulence, and shows a variable clinical expression depending on the status of the colony. This slide presents a clinical case of EFB characterized by a patchy brood pattern, the presence of dead or dying larvae in the unsealed cells. To combat Fouldbrood diseases, antibiotics are used, but in the European Union they are banned, including for prophylactic purposes. In the case of AFB, honeybee colonies must be destroyed under European legislation, as treatment is not permitted. EFB can be treated. Preventing Fouldbrood diseases through the use of plant extracts with antibacterial activity as an alternative to prohibited antibiotic therapy is essential for sustainable beekeeping. The present study aims to test in vivo and in vitro antibacterial activity of the herbal product BACTEROMAT herb produced by Extract Pharma, Sofia, Bulgaria. To achieve this goal, we conducted two types of experiments, laboratory tests in vitro and field studies in vivo. In vitro antimicrobial activity was tested using the agar diffusion method on P. larvae and M. plutonius with sterile water as a control. Cultures were incubated aerobically and anaerobically. After the lab test, we moved to field conditions studying the product's effects on naturally infected bee colonies. Two apiaries were included in the experiment. Apiary 1 comprised 130 bee colonies, of which 9 showed clinical signs of European Fouldbrood. Apiary 2 comprised 27 colonies, with 5 showing clinical signs of EFB. The bee families were housed in 10-frame root hives, free from symptoms of other diseases at the beginning of the experiment. Diagnosis of EFB was performed at the National Reference Laboratory for bee diseases following accredited methods. Field challenge was conducted under real management conditions, and samples were collected in both apiaries by veterinarians servicing the apiaries. The herbal product BACTEROMAT herb can be applied both on its own and in combination with other herbal products that stimulate the immune system of honeybees. You can see the treatment protocol combining BACTEROMAT herb with Immunostar herb applied 6 times at the specific intervals. The product offers different methods of application depending on the purpose of prevention or treatment, by spraying, by dripping, and with syrup in the feeders. Now we move to the laboratory results. The measured inhibition zone demonstrates the antimicrobial activity of BACTEROMAT herb. P. larvae showed strong inhibition at 1 to 10 dilution, and plutonius exhibited inhibition in both solubilities with smaller zone than P. larvae, indicating pathogen-specific differences in sensitivity. Following the lab result, the field data show rapid improvement in larva health just 1 month after starting treatment. We found that initially patchy brood pattern transitioned to denser brood. Healthy brood architecture by day 75 with progressive colony stabilization. In apiary 1, occasional patchy brood was observed without clinical signs of European Fouldbrood. Preventive treatment was carried out in the following season, and no winter mortality was recorded through the observation period. No clinical signs of EFB were observed during the 2 years following treatment. Here are examples for the first apiary showing the visual evolution of the brood after product application in 2021. This series of photos illustrates the transition from infected to healthy brood, allowing you to follow the recovery step by step. The first photo was taken in May and shows spotty brood with diseased larvae. The second photo of the same frame was taken in August after the application of BACTEROMAT herb. It clearly shows spotty brood without diseased larvae. The next photo was taken in late August during continued treatment. It shows slightly spotty brood without diseased larvae. The following photo was taken before the colony was winterized in September. It shows normal brood without diseased larvae. The last photo was taken in February of the following year, showing the already overwintered solid and healthy brood. In apiary 2, 1 month after treatment, no newly infected larvae were observed. The colony showed sustained clinical recovery following the initial treatments, and the families were well prepared for the winter. No winter mortality occurred through the observation period, and in the next 2 years, no clinical signs of European Fouldbrood were detected. The photo shows one of the most heavily infected colonies in apiary 2, displaying the classic signs of European Fouldbrood. The brood is spotty, with dead or dying larvae in unsealed cells. Many larvae are twisted or abnormally positioned, and their color range from pearly white to yellowish or brown. The summary of the field application of BACTEROMAT herb under field conditions in colonies affected by European Fouldbrood is as follows: absence of EFB recurrence post-treatment, stable overwintering performance, field evidence consistent with laboratory trends. We present the opinion of the owner of Apiary 1 regarding the application of BACTEROMAT herb and the results obtained. According to his opinion and years of experience using the product, both alone and in combination with other herbal products, BACTEROMAT herb has proven to be an effective alternative to antibiotic treatment. Plant extracts support bee health and control pathogens, with ethanolic extracts generally showing stronger in vitro effect due to higher content of active compounds. BACTEROMAT herb contains active herbal compounds as flavonoids, polyphenols, essential oils, vitamins, and minerals. It inhibits P. larvae more strongly than M. plutonius in vitro and can stimulate the immune system in vivo. Spraying sugar syrup supplemented with BACTEROMAT herb onto frames gave better results than feeding. BACTEROMAT herb can be used both alone and in combination with other immune-stimulating herbal products as part of an integrated strategy for EFB control. Combining herbal products at specific intervals enhance their effect, even in mixed infections with bacterial pathogens and parasites as Nosema serrani. While lab results may not always reflect field conditions, in vivo tests in apiaries with confirmed European Fouldbrood showed that 6 treatments with BACTEROMAT herb and Immunostar herb eliminated clinical signs, and 3 years of monitoring confirmed no relapses, demonstrating practical efficacy. The practical implications show that these products can reduce antibiotic use and be integrated into preventive strategies. They are also safe for bees and do not contaminate bee products. In conclusion, BACTEROMAT herb, especially in combination with Immunostar herb, effectively controls Fouldbrood and offers a sustainable alternative to antibiotics. Finally, we thank all partners, laboratories, and beekeepers who made this study possible. Details about the publication and access to the full text can be found below. Thank you for your attention.